Hello everybody, my name is Antonia Santos. I am a sophomore biology major at Hampton University from Chicago, Illinois.
My name is Asha Hawkins Rivers. I'm also a sophomore biology major at Hampton University from Lansing, Michigan.
And today we'll be discussing common sea stars of the Atlantic Ocean and Chesapeake Bay, the Asterias forbesi and the Asterias rubens, and their various importance. So to begin, when one stops to think about sea stars, which we fondly and mistakenly call starfish, the first thing that may come to mind is a familiar and lovable cartoon character that we all know. We do not stop to think about the reality of the animal. What is a sea star? Where and how does this animal live? And more importantly, why should we care about this creature? Well, to begin, the sea star is a member of the Asteroidea within the phylum Echinodermata. The Echinodermata are the sister group to the Xenoturbella and Hemichordates. And echinoderms are a very interesting group. Included within the phylum are the classes Asteroidea, which are the sea stars as we mentioned, the Ophiuroidea, which are brittle stars and basket stars, the Echinoidea, which consists of the sea urchins and sand dollars, and the Holothuriidea, which are sea cucumbers. The Asteroidea is one of the largest, most diverse, and species-rich class within the phylum Echinodermata. They occur all across the world from Scotland to Brazil and are very common with the Atlantic and Indo-Pacific regions. Asterias is a genus within class Asteroidea, and within this genus are the most commonly found sea stars, Asterias forbesi and Asterias rubens. Like many typical sea stars, Asterias forbesi and Asterias rubens demonstrate the typical morphological characteristics, including pentaradial symmetry, 5 or more rays connected to a central disc, ambulacra and ambulacral grooves, ossicles, and madreporites, and components of a water vascular system. Asterias forbesi and Asterias rubens are dioecious, meaning that there are separate sexes, and their fertilization is external. Their life cycles consist of a distinct bipinnarial larva stage, which many of us have seen if we've taken a biology laboratory course. While Asterias forbesi and Asterias rubens have not contributed significantly to the field of medicine, studies have been conducted with relatives of the two species that show promising innovations in the field of stem cell research and pharmacology. In the field of molecular biology and genetics, extensive research has been conducted using various macromolecules found within Asterias forbesi and/or Asterias rubens. These macromolecules play a variety of roles and perform many functions from protein degradation, concentration, and function to defense mechanisms utilized by sea stars. The research conducted regarding the molecular biology of sea stars has revealed that many macromolecules and defense mechanisms have been evolutionarily preserved and can be found, or their analogs can be found, within vertebrate and mammals. The ecological and commercial importance of sea stars is also of great concern due to their relationship with bivalves in the Chesapeake Bay and the Atlantic Ocean. The food chain involving sea stars and bivalves immensely impacts the ecology of the area.
Sea stars are found in benthic marine areas, which means they live on the bottom of the ocean floor. They're also found along shorelines and among coral reefs and rocks. They attach themselves to the coral reefs and the rocks with their tube feet to hold on during strong currents. They live in these areas of high salinity because they cannot osmoregulate. Osmoregulation is the ability to control the water flow in and out of your cells. But due to the fact that sea stars cannot do this, they're called osmoconformers, which means they've made a way for themselves to be able to osmoregulate without actually doing it.
Okay, now for a brief introduction into the taxonomy, phylogeny, and evolutionary placement of Asterias. So Asterias is a genus within the family Asteroideae, and of course it is within the phylum Echinodermata. Studies speculate that Asterias rubens and Asterias forbesi, the two most common sea stars, diverged into two separate species approximately 3 million years ago. Unfortunately, there are very few fossil records to support this hypothesis due to the fact that the skeletons are rarely preserved due to their lack of rigidity, and they tend to rapidly disintegrate. There are two hypotheses that discuss the placement of echinoderms in evolutionary history. One hypothesis states that the class Asteroidea is evolutionarily and phylogenetically placed as a sister taxon to Ophiuroidea, Echinoidea, and the Holothuriidea. However, the opposing hypothesis states that the Asteroidea are the sister taxon only to the Ophiuroidea and are basal to the Echinoidea and Holothuriidea. One hypothesis suggests that if Ophiuroidea and Asteroidea were to form a clade, then some morphological features, such as ambulacral grooves, had to have been lost in Asteroidea or have evolved separately in Ophiuroidea. Okay, now for a brief overview of the morphology of Asterias. So Asterias, like other typical sea stars, possess pentaradial symmetry and typically have 5 or more rays merged with the central disk. The oral side of the body possesses the mouth and bands called ambulacra, which extend from the mouth to the tips of each arm. The ambulacra possesses the characteristic tube feet within the ambulacral grooves. Ossicles cover the exposed nerves within the umbilical grooves, and on the arboreal side of the body are tiny pedicellaria utilized in keeping the organism free from algae and debris. Papillae, an anus, and the madreporiae are also present. Asterias forbesi and Asterias rubens can range in size from 5 to 15 centimeters in diameter and are typically neutral colored with undertones of red, orange, or pink. Asterias rubens can slightly differ with undertones of purple as well. The spiny ossicles are also a large and noticeable component of Asterias forbesi. In Asterias forbesi, the madreporiae is typically pink in color. Like most echinoderms, Asterias utilize a water vascular system for purposes such as locomotion, respiration, and excretion.
Now I'm going to go into the general behaviors of sea stars. Sea stars lack cephalization. However, some sensory organs are present, such as a nerve plexus and eye spots at the end of each arm. Asterias forbesi relies heavily on chemoreception to orient themselves and get around. They use this sensory technique to locate prey and predators by the ocean's current sweeping the odor over the sea star. They use the water vascular system and their tube feet to locomote to either get their prey or run away from their predators. As the water vascular system pumps the water pressure to from one arm to another, the tube feet release and regrasp onto a stable surface. Despite their pinwheel shape, sea stars do not go in a wagon wheel formation when they move from eye spot to eye spot. However, they move in straight lines and angles across the ocean floor. If a sea star gets flipped over by a strong current or a passing by animal, they will push the water pressure into one arm so that it lifts up into the air, and they will keep exerting the water pressure until they basically do a somersault and land on all 5 arms. Now I'm going to give a brief overview of the ecological interactions of sea stars. Sea stars are carnivores that prey on mollusks, crustaceans, and small organisms such as plankton. The observed preference of the Asterias forbesi are bivalves such as oysters, clams, and mussels. The sea star captures the bivalves with its arms and uses its tube feet to apply pressure to the bivalve shell. The adductor muscles eventually loosen enough so that the sea star can inject its stomach into the bivalve and consume its nutrients, leaving an empty shell. Sometimes sea stars will dig for their prey in sand to get the bivalves that are really close to the surface, but they will not go deep into the bed of oysters or mussels that are often found on ocean floors. If a clam or an oyster is big enough, sea stars, even though they don't colonize, they will help each other and gang up on the oyster and defeat it together. There are not many commensal relationships with Asterias forbesi. However, there is one with the Caprelli grahami that was discovered many years ago. This species will cling to the back of the starfish in between the spicules with its posterior periopods and eat the floating by nutrients and plankton that graze the top of the starfish.
Okay, now we're going to briefly discuss the molecular biology of Asterias forbesi and Asterias rubens. We're going to briefly talk about a few macromolecules that are of significance within these organisms. The first is the macromolecule called a steroid glycoside. So, to begin, while the molecular biology of any living organism is complex, unique, and amazing in its own way, much research has been conducted concerning the molecular biology of the Asteriadea. So, the glycosides are also called the glycosides of polyhydroxide steroids, which is quite a mouthful. These molecules are of interest to many scientists in that they have been found to have numerous abilities and effects. They are reported to possess antifungal, antiviral, and anti-inflammatory effects and other abilities. Polyhydroxide steroid glycosides in Asteriadea function as a cytotoxic compound that reportedly influence cytotoxicity and hemolytic activity. The next macromolecule we'll discuss is called capsin D. So to begin, proteolytic degradation, or proteolysis, is the degrading of proteins and plays a key role in the protein concentration and function in many organisms. Proteolysis is done through a number of pathways, one of which, the one that we are interested in, is in relation to capsin D and the lysosomal pathway. Thus, capsin D is a lysosomal enzyme that plays a role in proteolysis in sea stars. It is of particular interest primarily because capsin D has been isolated in other vertebrates and invertebrates, and it is strongly believed that it has been evolutionarily conserved from older phyla up to mammals. And finally, the last macromolecule we discuss is nitric oxide. So vertebrate phagocytes are capable of producing various molecules utilized in host defense. One of the most forceful is nitric oxide. So it was recently discovered that invertebrate phagocytes are also capable of producing reactive nitric oxide. This was accomplished through a study by Gregory Beck. Seeing as to how coelomocytes from invertebrates have similar functions to the phagocytes, Beck used these cells, which were obtained from Asterias forbesi, in his study. So this was done through the investigation of the effect of various stimulators on nitric oxide production. The data collected concluded that coelomocytes were indeed capable of producing and releasing nitric oxide. So this confirmed that nitric oxide is a chemical mediator that has been evolutionarily conserved as a defensive weapon. Finally, I would like to discuss the medical importance of Asterias forbesi and Asterias rubens, or just sea stars in general. So, as we all know, the regenerative properties of sea stars are of great medical importance, and especially in the area of stem cell research. So basically, we all know that when a sea star loses an arm, it can basically regrow it. This is of interest to us as humans because we can't regrow our arms. So we all know that stem cells are cells that are capable of self-renewal and differentiation. By studying the mechanisms with which sea stars are able to regenerate their limbs, researchers are given a glimpse into the regenerative and healing capabilities in humans, and thus we can find new and innovative ways to use stem cells. Also of importance and of interest is the discovery that a slime produced by a specific type of sea star may have an impact on inflammatory diseases such as asthma and arthritis. So while the sea star of interest in this case is not Asterias forbesi or Asterias rubens, it is a relative of theirs called the Marthasterias glacialis, a sea star found near Scotland and the British Isles. So a slime that is produced by these sea stars could be vital in treating the above mentioned diseases, because the slime is a defense mechanism that prevents debris from adhering and accumulating on the sea star. So a purified and synthetic version of this slime can potentially be used in blood vessels to prevent white blood cells from sticking to and building up within the walls of the blood vessels. So research is currently being done to determine whether the slime can be modified and synthesized into a drug that we can use to coat the walls of blood vessels to aid in the treatment of such inflammatory diseases. Well, this is the end of our presentation, and we hope that we have given you a better understanding and a glimpse into the lives, the importance, and just the overall overview, I said that, of sea stars, specifically the Asterias forbesi and the Asterias rubens within the Chesapeake Bay and Atlantic regions.
Thank you for watching.